Hello everyone. I welcome you to CAR Journal Club, which we conduct every month. CAR has been founded by Dr. Pratibha Shah. It is a pioneering initiative to promote and establish Ayurveda as an evidence-based health science globally. And one of our core goals is to create a platform for the exchange, interaction, and integration of information, competencies, expertise, and resources between Apex academic institutions, research practitioners, students in USA as well as in other countries globally with highest global standards. And our long-term goal is to become the nodal agency for Indo-US collaborations and initiatives for research in Ayurveda, with ultimate aim of establishing Ayurveda globally as a credible healing science. We are headquartered in USA, but we are expanding our visions to other countries. And I invite you all to join our team as members, volunteers, and for organizations, institutions as for collaboration, locally as well as globally. Please check our website, ayurvedaresearchusa.org. Today, our presenter is Dr. Ritika Shah. She is an Ayurveda consultant and she has completed her graduation and MD in Ayurveda from D.Y. Patil University, Navi Mumbai, India. She contributed as a frontline doctor in India by treating asymptomatic COVID patients with Ayurveda. She is also the founder of an Ayurvedic brand named Herbostra. She has immense faith in Ayurveda, but she is also well updated with the latest developments in medical science. Today in the Journal Club, she is going to present a published research titled G14A, a polysaccharide from Tinospora cordifolia, inhibits the survival of Mycobacterium tuberculosis by modulating host immune response in TLR4 dependent manner. So over to you, Dr. Ritika. You can start your presentation now. Thank you very much for being with us and presenting.
Thank you so much, Dr. Vandana. Good evening, everyone. I'll quickly share my screen. So today for the Journal Club meeting, we'll be discussing a research paper, G14A polysaccharide from Tinospora cordifolia inhibits the survival of Mycobacterium tuberculosis by modulating host immune response in TLR4 dependent manner. So this paper was published in PLOS ONE journal, PLOS ONE or Public Library of Science journal. It was published on May 5th, 2016. It was cited 31 times. The study was conducted at Baba Atomic Research Center in Mumbai, India. And it was written by Pramod K. Gupta, Pampi Chakrabarti, Santosh Kumar, Pramila K. Singh, MG K. Rajan, Krishna B. Sainis, and Savita Kulkarni. So this is a disclaimer. This presentation is for academic purpose to understand methods for critically appraising a scientific manuscript. It is by no means intends to harm or criticize the investigators or any other personnel connected to the manuscript or clinical study. The views expressed herewith are with sole purpose to be academic. The presenter declares no conflict of interest. So the reason why I chose this paper is because this paper has anti-tuberculosis activity. TB still is a global health concern. It is caused by Mycobacterium tuberculosis. It is highly infectious and it is an airborne disease. So every 22 seconds, a TB patient dies. So around the statistics for 2019 were 1.4 million in a year, which is too much. And due to the pandemic, the condition has become even more worse. And it is estimated that in the next 5 years, additional to the 1.5 million per year, there'll be 6.3 million more deaths. So, and the combination of COVID and TB is a deadly combination. It's going to make even more worse. And the fight against TB is going to make even more difficult. So the reason that we have no control over the disease and the bacterial burden is even still high is because of n number of reasons. One is the patient non-compliance to the long-term therapy, which the first line of anti-TB drugs have, emergence of MDR, extensive drug resistance, then the adverse effects which the anti-TB drugs have on the patient, and the degradation of the drug before reaching their target are the main disadvantages of the conventional treatment which is available right now. So this is one of the thrust areas in research, if we call it anti-TB activity. So we have seen that there are a lot of Ayurveda medicinal plants which have shown a lot of good results in the treatment of TB. So there's a lot of scope for Ayurveda medicinal plants to be used in the treatment of TB. Hence, and even though simultaneously there are a lot of development of antibiotics, but that it's very slow and it's going to take years to develop a new antibiotic for TB, and the drug resistance is not going to stop. So, and one of the main other reason is I was going through a lot of anti-tuberculosis researches of medicinal plants, and most of the medicinal plants had anti-tubercular activity was shown in vitro studies, where obviously a lot of medicinal plants have showed antimicrobial activity, but that's not the only way Ayurveda herbs work. Ayurveda herbs or medicinal plants are multifaceted. It works in various different ways and has a lot of different properties. For example, guruchi, here the property of antibacterial property of guruchi was seen, but also immunomodulatory activity was also seen, which correlates with balya and rasayan property of guruchi. So one of the major reason of we have no control over the disease, or the reason for MDR drug resistance, is the ability of the Mycobacterium tuberculosis to adapt to the changing host environment. The Mycobacterium tuberculosis, the main pathogen which causes tuberculosis, it has the ability to adapt to the changing host of environment and parasite the host, the whole macrophages, which is the first line of defense of immunity. It parasites that macrophages and inhibits the immune response, which is directed against it. So it inhibits a lot of immune response functions, which are fusion of phagosome, lysosome, antigen processing, reactive nitrogen intermediates, host cell apoptosis, and responsiveness of IFN gamma. So even though there are a lot of effective treatment for TB right now, but still people are getting resistance and the TB burden has not decreased. So there has to be something new and novel strategy has to be applied for the treatment of TB, because this is definitely not that effective. So the one of the main things symptoms we've seen is the innate immune response in tuberculosis is not good. It is compromised. So we can get drugs which boost that immune response to get a better treatment. So G14A is one such they've taken in the study. It is a polysaccharide which is isolated from Tinospora cordifolia, which is also known as guruchi in Ayurveda. So their hypothesis here is that G14A inhibits the survival of drug-sensitive uh, as well as multi-drug resistance and Mycobacterium tuberculosis in vitro macrophage model in TB infection by inducing nitric oxide in TLR4 dependent manner with simultaneously induction of pro-inflammatory cytokines. So G1-4A is a polysaccharide which is found in Tinospora cordifolia. This is a biopolymer. It is a starch-like chemical which is found in plants usually attached to proteins. They are commonly used in plant growth. They are used in pharma industries for binding, for its gumming and binding agent. And they are also said to have immunostimulating activity. So that's what, so this particular G1-4A is a polysaccharide found in Tinospora cordifolia, and its extraction is patented. So we're going to study the immune response. So this kind of study where immunomodulation is studied while inhibiting the growth of the bacteria or checking a certain activity. So this is usually seen in This is usually these studies were also seen in anti-cancerous studies where they are trying to study the immunomodulation. So first we'll see how Mycobacterium tuberculosis inhibits the or compromises our immunity. So this is the normal activation of macrophages in innate immunity. So whenever a pathogen enters the body, it enters the M1 pathway and activates the macrophages, which leads to a lot of increase in cytokines, which is TNF alpha, IL-1 beta, IFN gamma, interleukin 12, and interleukin 6, which in turn enhances the production of nitric oxide, which stimulates the T cells in the body, and it upregulates the MHC and CD86 molecules, which in turn stimulates the microbicidal property, which helps in phagocytosis. This is the normal activation of macrophages. When a Mycobacterium tuberculosis pathogen enters the body, it alters the activation of macrophages. It completely skips the M1 pathway and takes an alternate pathway, which is the M2 pathway. Due to this, there is no production of TNF alpha, of all the cytokines, IL-6, IL-1, which is mainly the first line of defense of immunity. So this in turn inhibits phagocytosis and it decreases MHC and CDC. One thing in the M, when it takes an alternate pathway, it increases the interleukin 10 cytokines, which has an anti-inflammatory effect, which inhibits phagocytosis. So this makes macrophages uh, let the Mycobacterium tuberculosis survive in our body, so which in future leads to a chronic infection. So we'll study the methodology, what exactly here the author has done. First, they've isolated G1-4A from Guruchi. So then the study is divided into two parts. One is the cell line study of macrophages, and the other is the animal study. In the cell line study, different strains were used and divided into two parts. One is the uninfected, and the other is infected strains. The uninfected strains, one was treated with G1-4A, and one control group was kept in the absence of G1-4A. In the infected one, three types of strains were taken. H37RB, which is a standard tuberculosis strain. Then two different MDR strains were taken. One is Beijing and one is Lamb. The reason these two MDR strains were taken were because they are responsible for the maximum number of MDR uh, infection. That's why these two strains were taken. And in turn, these two strains were divided into two groups, one which was treated with G1-4A and the other ones which are not treated with G1-4A. The next is the animal study, where 6 weeks female mice were taken, 6 in each group, and aerosol infection. They were infected with Mycobacterium tuberculosis. The same thing, there were two groups were divided in two parts. One is uninfected, infected, one which was treated with G1/4A, one in the absence of G1/4A. The infected ones, three strains were taken, the H37RV, MDR Beijing and MDR Lamb, and both in absence, and they were treated with G1/4A. In these animal study, three things were observed. One, RT-PCR of left lung was taken to check whether the Mycobacterium tuberculosis survives in the body. Then the levels of cytokines were observed to see the immunomodulation and just to see the intracellular activity of G1/4A. And another study was taken as an adjuvant therapy along with a first line drug, isoniazid. So these are studies. Uh, so there are a list of assays which was uh, these were the list of assays which we won't go much in detail because we'll focus on the results here. So this was the first cell line study where the levels of cytokines on cell line were studied. So if you see in the first graph A, the IFN gamma, which is responsible for, it's part of the first line of defense of our immunity. If you see the uninfected, untreated one, the concentration of IFN gamma is very low, but the one treated with G1/4A has a significant increase. The same goes, one was the one infected with H37RV. The if you see the untreated one is around 5, and there's a significant increase in the ones which is treated with G1/4A. The same, you see the similar significance in MDR Lamb and Beijing. So there has been a constant increase in IFN gamma, interleukin 1 beta, interleukin 6, and TNF alpha. If you see the D1, the last one, TNF alpha, there's a constant increase in all four strains, the uninfected, H37RV, Lamb, Beijing. This is interesting because TNF alpha is responsible for cell signaling in our body. So whenever there's a pathogen when it enters the body, the cell has to signal the rest of the body that there's a pathogen in the body, which TNF alpha does. It's a function of TNF alpha cell signaling. So there's a constant rise in that, which is a very positive result. The next is the secretion of nitric oxide and NOS2. So nitric oxide plays an important role in neurotransmission, vascular regulation, immune response. And NOS2, nitric oxide synthesis 2, is responsible for the function of immunity against various viruses, bacteria, and fungi. So even here we see the cell line, which is untreated is really low and has a significant increase when it's treated with G1/4A. In both the first one, nitrite and NOS2, they stimulate the TLR4 function and makes the first line of defense much more stronger. Further, the phagocytic action of so all the cytokinins have been stimulated. Now these cytokinins have to go and actually kill the bacteria. So here the phagocytic action is seen. So if you see the uninfected has around 5%, untreated is also 30%, while the one treated with G1/4A has 65% of action, which is more than half of action. So these are CD86 and MHC cells. So these are also responsible for immunity. And MHC cells are even more important because it helps in recognition of a foreign substance in the body. So here also we've seen that there is a drastic rise in MHC cells and CD86 cells, which of the strains which were treated with G14A. Yeah, this interleukin 10 is in the M2 alternative pathway. It is secreted there and is responsible for anti-inflammatory activity. So whenever a pathogen enters the body in the M2 pathway, interleukin 10 rises. So if you see the cell line which is infected with HT7 has around 98. Uh, so this is usually seen in chronic infection. That's why the study has been observed in 15 days, 30 days, and 60 days. So in 15 days, there's not much difference, but on 30 days, it's at the peak. So the one treated with G14A has a sudden decline in the levels of IL-10. And 60 days, because it's a protective action, it declines. This is the intracellular survival of MTB strains, which basically shows that it inhibits the activity of Mycobacterium tuberculosis. This is the same thing. The colony forming units in the study has declined drastically in the strains which were treated with G14A, and the ones which are not treated had increased over time, so which have been observed in 48 hours. So the cell line was treated with G14A 8 hours prior to the infection. Then after the infection, it was treated for 48 hours. And then it was observed after 48 hours and 72 hours. So if you observe the B graph, the bacterial burden in the control group is 100. In the ones treated with G14A, there's a sudden decline in the bacterial burden, which is around 30 to 40%. And after 72 hours, it further declines. So then in the animal study, uh, the lungs from the mice were removed and they were observed. In this study, they have also taken INH, isoniazid, which is the first line anti-TB drug, in consideration, and the results were compared. So if you observe, the untreated cells were obviously the bacterial burden observed was much more. INH has a sudden decline. But if you see the last one, G14A plus INH, the bacterial burden has decreased when both of them were treated together with G14A INH. So one of their results have also shown that if G14A is used as an adjuvant therapy along with the first line of drugs, the results achieved will be much more. So these are the results. G14A modulates the host protective immune response in macrophages infected with drug-sensitive and MDR strains of Mycobacterium tuberculosis. G14A treatment augments the phagocytosis of MTB in murine macrophages, which we saw that the phagocytic action, does have phagocytic action. Then G14A inhibits the intracellular survival of drug-sensitive and MDR strains of MTB in macrophages, partially through nitric oxide production in TLR dependent manner. This is also we've observed that stimulates the production of nitric oxide, which in turn stimulates the TLR4, which is our first line of defense of immunity. G1/4A inhibits the survival of MTB in mice lungs, and when given in combination with INH, compared to isoniazid alone and G1/4A alone, the activity of both of them together is much more compared to their activity alone. So G1/4A has shown effect in all the three strains, the standard strain and the drug resistance. So cell-mediated immunity is critical to control and eradication of Mycobacterium tuberculosis. So this could be a potential adjuvant therapy for tuberculosis, but for this, a lot more study is still has to be still taken because this is not enough. Clinical trials, more animal studies, more in vitro studies has to be conducted to prove this hypothesis. So it's a really good study. I was really impressed of what they've done. They've it's a quite detailed study, but few things which they were not clear. One is the use of Middlebrook medium for isolation of Mycobacterium tuberculosis. Mycobacterium tuberculosis, you can use different mediums. You can use LG medium and MB-BACT, but usually MB-BACT medium is preferred in isolation of MTB. So there's a research study also which compares all three of them, but I don't know, they've not mentioned why they use Middlebrook medium for isolation. This does affect the accuracy of the results and contamination, if there's any, in the strains. Then the concentration of G1/4A in the study is not mentioned. Whatever, even in the cell line studies and even in the animal study, they've not mentioned that at what concentration G1/4A was used. This is important because especially when you're comparing it with a drug, INH, which if you go to see the minimum inhibitory concentration, MIC of INH is 1 μg per ml, which is comparatively the lowest you can see in an anti-TB drug. And if I did found a few research on Tinospora cordifolia, and the MIC for Tinospora cordifolia is 50 μg, which is 50 times INH. So, the concentration of G1/4A used in the study is very important. They should have mentioned, because the results must have been totally different when used in the right concentration. Then they've mentioned that they've taken mean results from 3 independent experiments of each strain, but they've not mentioned much details. I only saw that a small star note which is mentioned below the graphs. So there are not much details about that. There's a lot of technical data in the study, but the groups which they took for animal study were not very mentioned clearly. How many groups they took, and was there any control group? While discussing, we do come to know what kind of groups were taken, but it's not very clear. Then the cell line study didn't have a control group with INH. If they did it for animal study, they should have done it for in the cell line study also, which would have helped us to compare the activity with Tinospora cordifolia. There was not much detail mentioned about the herbal drug Tinospora cordifolia or about G1/4A. There has to be why they chose G1/4A for this particular study. There was not much mention. There is a small mention of the experiment they carried out with LPS, that's lipopolysaccharide, and G1/4A, which needs more explanation because it showed that both of the activities were same and both of them inhibited the growth of the bacteria. So it needs more explanation, but I couldn't find it in the paper. And the same, the use of G1/4A is very new. Most studies have to be carried out to actually use it in a conventional treatment. Okay. Okay. that's it from my side. Thank you.
Thank you, Dr. Ritika. Very nice presentation. Um, if you like, you can stop sharing so that we can, see each other.
Yes. Um,
Thank you. That was a very complex study and presented very well. Good summarization, and really you brought out some gaps in the study that could have been probably conducted better. With that, I would like to open up for discussion, for comments. Anybody? You can please come on the video, unmute.
Hey, hi, Dr. Ritika. It was really nice presentation, and you presented very well because I couldn't go through the study before, but I could get everything. So it's very nice. One thing comes in my mind, like when you talk about the animal study, they said they took the tuberculosis. It says like right lung affected more easily, like bacteria. Like when we see the like comparative study, right lung mostly affected, right? And upper lobe and right lung. But in clinical study, they chose the left lung of the mice. So if they like put any clearation, why they took the left lung?
yeah, that's what they've not mentioned anything. In
Okay.
fact, that's why I said that they've not given a detailed description of what kind of groups they've taken. They've just mentioned that they did so and so thing, but a detailed description of animal study and what groups were taken and why only left lung was taken is not mentioned.
Yeah. Okay, thank you.
Wonderful presentation, Dr. Ritika. And you have chosen a very interesting study in the way that usually we see here clinical papers being selected, and in-depth study about an active principle for one of the Ayurvedic herbs is very interesting to see Ayurvedic pharmacology being proved through experiment and clinical research of this kind. Because in Ayurveda, Guruji has already been described Rasayan, Balya, Krimigna, Bhutagna, and say Vishagna. And it is just establishing that fact that it is antibacterial for such a common and devastating disease. And like you mentioned, they have, before our starting the paper, during the discussion, they have not acknowledged the herb to be Ayurveda herb and how it has been mentioned in Ayurveda for various properties, Ayurvedic properties, how it is used for health and protection of health, and not just that, improving the quality of our health. So that is also a lacune, I would say.
True, true. That's what they've not mentioned. If they could have correlated with Ayurveda, it would have been a really good study, or much more better study if you will say. It is a good study.
Yes. And
Yeah.
did you find any reason why they chose this particular herb for Mycobacterium?
No, but I have seen there are a lot of studies on G1/4A which talk about immunomodulation. So they needed one, and Tinospora, we know it is most researched drug in Ayurveda.
So based on
So
that, it might
I
have
think they must
influence
have,
there.
yeah.
Good to know that. Thank you. Anybody from our who have joined, Pratiksha, Vineet, and Kishore, thank you for joining. If you have any questions, you can put comment or you can switch on your video. You are welcome to join.
Thanks a lot. Thanks, Ritika. Sorry.
Yeah, go ahead.
Continue. Sorry.
Thanks, Ritika.
Go ahead.
Sorry.
Maybe Kishore, you want to go ahead first?
No, okay. Continue. I'll speak later. Thank you.
Sure.
I just wanted to point out one thing. Thank you, Dr. Ritika. It was really nice. I mean, someone from a non-medical background, this was really astonishing to know more about tuberculosis and how Ayurvedic medicines would help.
Thank you for joining, Vineet.
Sure, my pleasure.
Kishore, would you like to share your comment?
No, thanks a lot. Thanks, Ritika. So very good information. Thanks you.
Are you related?
Yeah, that's my dad. And Vinith is my husband.
Oh, fantastic.
We in Zandu, we had done research on Tinospora cordifolia 30 years before. How it works on immunity, on cancer. In Zandu R&D research, we have done a lot of research on Tinospora. I'm talking 30 years before.
Interesting, very interesting. And you know what surprises me, that lots of research has been going on in India, and I do not know the research you are saying for Jandu. I am sure that it was in Indian labs. I do not see any papers or any publication that has come to global platform to validate any of these things for which we use these Ayurveda herbs. And we still are so back in our research where Ayurveda is concerned.
Yeah.
That is really very disheartening to see. When you say it has been going on for last 30 years at Jandu.
When Zandu was there, now it has been taken over by Imami. So before this is
Imami, yes.
A lot of studies we've done it on this, and it was
Are
surprising.
there any publications?
I think it was.
So Dr. K.M. Parekh, he was one of the, so he has a lot of research papers. He was the director in Zandu. So he inspired a lot of research papers on Guruchi. So that's why it said that, that was 30 years back. So Guruchi was the main drug for research even in US and even in KEM. A lot of research that's still going on Guruchi and KEM, anti-cancerous activity, a lot of tuberculosis, and a lot of other diseases. Also, we know that, it is one of the most potent drug. Even on COVID, in fact, we've seen so many researches.
Yes, its name is Amrita. See, simply the name is Amrita. It is Amritulya.
So where are the publications, Ritika?
I can find it online. Actually, it's not with me. That was an old research, but I can talk to Dad and
It'll be nice. We can add it to the repository that we have.
Oh, sure. I'll
definitely.
Find out.
I will look into it definitely and share it with you.
So I think I was intrigued to see that the site of the research was Baba Atomic Research Center, because to the best of my memory, maybe 25 years ago, they had done some kind of study on some bhasmas. And some paper was published on kind of the nanomedicine of Ayurveda.
Yes.
Yeah, but after that, I don't think I have seen any. So it's interesting that you have brought a paper that was, the study was conducted and published by non-Ayurvedic people at a non-Ayurvedic site. And if you actually read the abstract, and I'm sure you have read it in detail, it shows in your presentation, but it says Indian system of medicine, something, it mentions just that one word is mentioned. But considering that this has been conducted by non-Ayurvedic professionals, I think we have to give them some slack there, that, obviously they're not familiar with Rasayana and this and that. Effects of Guruchi So it's not surprising that they have not mentioned, but it is a very good crossover study, I would say. Maybe no, crossover may be the wrong word, but they have applied modern microbiology principles. And am I audible? Am I
Yes,
visible? Okay.
Yes, yes.
for a few seconds you were not. You're fine now.
Okay. Yeah, so for that reason, I think it's a very unique and interesting study that it's very purely microbiology intense. And, that side of research has been very elaborately conducted and reported. But there is value in whatever way they have done, and from here on we can pick up some of the data, and this can call for another 50 such studies, maybe, with more reasoning also why just this um, active principle was picked up, versus there are so many alkaloids, so many other active principles in Guduchi. Why this versus that, and why not the whole plant? Because in Ayurveda, those things will come in. We don't use single plants. Usually, we use polyherbals, etc., etc. But still, I think the study, despite all of the gaps that you have highlighted, I think it is a very unique study because it's very elaborate and it's some details are lacking, but still I would say very impressive study. And it's a statistically significant outcomes. The p-value was less than 0.05 in all of the 3 experiments.
Yeah.
Yeah. So still, I would say it can lead to a lot of new data, new directions. So that I think it's the strength. These are the strengths of this particular paper. Do you know which year this was conducted? Was it recent?
It was
2016.
2016.
Right, right. Okay. I was on the call in between, so I kind of missed a little bit. Okay. Any other comments or critiques? Very welcome. By the way, anyone who is attending Council for Ayurveda Research is kind of pioneering some lead in getting the research and world together. And we are actually, I can make this announcement. We are organizing a consortium, global consortium in September. This is our second global event. We have people from both sides of the industry and from people from the industry, actually, industry pharma, on the pre-conference day, which is Thursday. And then that will lead to a 3-day conference, which is already packed with some of the leading experts. All the speakers have been announced. And day 3 will lead to the formation of a consortium, which hopefully will become the body that will, give some new directions and some clarity, some consensus next steps for research in Ayurveda. There's so many research questions, including methodology. What should be the correct methodology? So, yeah. Any other final comments? How was your experience, Dr. Ritika, presenting in this probably new forum for you, right? Here in the
Yeah,
US.
yeah. So it was my first time. Good. The experience was good, and it is good to see so many Ayurvedic doctors doing such good work,
Yeah.
in Council of Ayurveda Research. So interesting. Still exploring.
Yes,
Okay.
yes.
Still getting to know the practice here. So,
Yeah, we've all walked that path. And this will be uploaded on YouTube, and we have just started our YouTube channel early this few months ago, and we already have 200 subscribers. So this will go on YouTube, and I will share with you also, Dr. Ritika. You can share it with people because everybody is not able to make it on a call on a Friday on a working day at this time. So people can watch it later. All right. Any final thoughts, comments? So journal club meetings are every month. So anybody who's listening, taking a listen later on also, you can check us out on Facebook page and our newly revived Instagram page under Dr. Ritika. So
Oh, so Ritika is taking
you
care of.
can-
Wow.
yes, I told you, no, Sharanya is sort of out because of the twins. So all right, then if there are no further comments, we are going to conclude for today.
Okay.
All right, thank you so much.
Thank you everyone for joining.
Thank you.
Thanks a lot.
Yeah,
Thank you.
thank you.
Thank
Thank you.
you.
Thank you.
Thank you.
Thank you.